Constanza.
And I am Pablo, and we are part of the Neurodevelopment Lab led by Carlos Oliva
In the Faculty of Biological Science of Casa Central Campus of UC.
We share the lab space with the Lab of Behavioral Genetics led by Dr. Campuzano. Most of our activities are done jointly in a nice and cooperatively working environment. In the Neurodevelopment Lab, we are interested in the development of the nervous system, focusing in the compartmentalization of the optic lobe and the signaling pathways involved in this process.
Some of the methodologies that we use include fluorescent microscopy of different developmental stages of the fly. To dissect this signaling pathway, we also use molecular biology techniques such as PCR, cloning, and bacterial transformation, Western blot, and primary cell culture of larvae neurons, and cell line cultures like S2 cells. To analyze expression patterns and mutant phenotypes that include morphology and connectivity defects, we use confocal microscopy. One of the key pathways controlling the development of nervous system is the slit-robo pathway, classically studied in context of axonal guidances. My PhD thesis project is focused in the autocrine slit-robo 3 signaling in this cells population. My focus is to characterize this pathway and its dependence of endocytosis.
I've been working in the role of the slit-robo pathway in the connectivity of two important neuron populations, photoreceptor cells and T4 and T5 neurons, which are part of the motion detection system. I'm currently working in my undergraduate thesis centered in the migration of neural progenitors that eventually become the neuroblasts that give rise to neurons of the lobula complex. Loss of function of slit mutants and ROBO3 mutants exhibit neuropil disruption and abnormal soma positioning.
Other research projects in our lab include adapting the Tango system to visualize the activation of serotoninergic synapses and evaluating the toxicity of copper in different fly tissues.
We are also developing a scientific kit to use Drosophila as a model for laboratory activities in schools. And that's it. See you at the Small Brains course.
Shall we start again? What do you wanna do then?